Intermediate filament cross-linking proteins play a very important role in cell physiology. Plectin is one of these proteins that can cross-link intermediate filaments. It can link the three cytoskeletal elements being intermediate filaments, microtubules, and actin filaments. So this protein, plectin, can bind these three cytoskeletal elements, and it plays a very important role in organizing the cytoskeleton. And as we know, the rest of the cellular organelles are also influenced by the position of these cytoskeletal elements. So let's take a look at this protein. Plectin binds intermediate filaments and links the intermediate filaments to microtubules, actin filament bundles, and filaments of motor protein myosin. By holding these different networks together, plectin plays an important role in maintaining the mechanical integrity and also viscoelastic properties of the tissue. Additionally, plectin helps in attaching intermediate filaments to adhesive structures at the plasma membrane. Here in this micrograph, you can see the different colors, and I'll tell you, plectin is green colored in this micrograph. Here's the green color, and the red is the microtubule, color of the microtubules. Microtubules are colored red in this micrograph. So plectin is here, the green filamentous structures you can see. These yellow dots are the antibodies which are specific to plectin, and these antibodies have been attached. A gold particle has been attached to these antibodies, so we can identify which structure our antibody is binding when we look at them under the electron microscope. The blue structure, this blue string, is intermediate filament. This intermediate filament is interacting with this green structure, which is plectin, and it is linking these filaments together. Also, I would like to mention that we don't see actin filaments, and I've said plectin can also bind intermediate filaments to actin filaments. The reason why we don't see actin filaments is because they were taken out when the sample was being prepared. If we had left the actin filaments in this tissue, we would not be able to see these interactions of plectin with other cytoskeletal elements. So here's a photograph of plectin binding intermediate filaments to the microtubules. Plectin gene mutations cause a devastating human disease that combines epidermolysis bullosa caused by disruption of skin keratin filaments. We have talked about this disease previously in a different context. Muscular dystrophy caused by disruption of Desmin filaments. Desmin's filaments are a type of intermediate filaments found in muscle cells. And neurodegeneration because of disruption of neurofilaments. We'll talk about neurofilaments later, but neurofilaments are a type of intermediate filament found in neuronal tissue. Mice lacking functional plectin die within days after birth with blistered skin and abnormal skeletal and heart muscles. This suggests that plectin may not be necessary for the initial formation and assembly of intermediate filaments. Its cross-linking action is required to provide cells with the strength they need to withstand the mechanical stress inherent to vertebrate life. Here we have micrographs of intermediate filaments that are found in neuronal tissue, in neurons, the type of cell which makes the nervous system. I'll explain this. Many intermediate filaments bundle themselves by self-association. Neurofilament proteins, NFM and NFH, contain C-terminal domain that extends outward from the surface of the assembled intermediate filament and binds to neighboring filament. Analogy would be if you were talking about microtubules, rather than tau attaching to the microtubule, imagine if the microtubule subunit had a little protein, a piece of protein or polypeptide chain that was sticking out in the space. And that protein segment of the monomer had the ability to bind microtubules which are lying adjacent to it. So these proteins which make intermediate filaments have special domains that radiate out of the filament, and they can bind other adjacent intermediate filaments. And since this happens in neurons, we call the intermediate filaments in neurons or neuronal cells the neurofilaments. Neurofilaments form robust parallel arrays that are held together by multiple lateral contacts, giving strength and stability to the long cell processes that neurons put out, axons and dendrites. The micrograph on your screen is freeze-etched electron microscopic image of neurofilaments in a nerve cell axon. You can see extensive cross-linking arrangement gives the cell process the great tensile strength. Here you can see these structures sticking laterally out of the intermediate filaments or neurofilaments. These are binding two different intermediate filaments. Here's filament number 1, here's the filament number 2, and one of these filaments is producing these lateral domains which are binding and hooking up with the filament which is lying adjacent to this one. In the other photograph, we have a freeze-etched image of glial filaments in the glial cells. The intermediate filaments, you would notice that are smooth and they do not have very many cross bridges. Flagrin is another protein that bundles keratin filaments in differentiating cells of epidermis to give the outermost layer of the skin their special toughness. Flagrin deficiency has also been linked to a more severe form of eczema, and also because of that, it persists into adult life. Here I would like to show you a photograph of two mice. The one on the top is a normal mouse. The one in the bottom has mutation in this gene, flagrin, and because of this, if you would notice, it has ichthyotic skin, which is it has a fish-like skin. It appears sort of scaly. So microtubule intermediate filament bundling proteins, they play a very important role in maintaining the cellular functions that allow cells to perform specific tasks, especially in neurons and also in the dermal tissue.